Presented by Caltech.
Good evening. I'm Jackie Barton, and I'm chair of the Division of Chemistry and Chemical Engineering here at Caltech, and I'm delighted to introduce our speaker this evening, Professor Linda Hsieh-Wilson, my colleague, and the Arthur and Marian Hanisch Memorial Professor of Chemistry. Linda is an organic chemist and a neurobiologist. She attended Yale University as an undergraduate, then obtained her PhD at UC Berkeley with Peter Schultz, an alumnus of Caltech, and then carried out postdoctoral research in neurobiology at Rockefeller with Paul Greengard. Linda is focused on research on sugars and more complex carbohydrates, and trying to understand how these sugars are used for signaling between molecules and signaling on the cell surface. It's hard to make sugars, and harder still to characterize them. And what Linda has really done is develop the tools to allow one to probe these signaling mechanisms on the surface of cells using her expertise in organic chemistry and in neurobiology. Based on this work, she's obtained many awards. She's a Sloan Fellow, a recipient of the Eli Lilly Award in Biochemistry from the American Chemical Society, and the Horace Isabelle Award in Carbohydrates, also from the ACS. Now, Linda actually wanted me to tell you, to be sure to tell you, that she's not going to be talking tonight on autism or neuropsychiatric diseases. Instead, she's going to be focused on her work on axon regeneration. I'm very excited to hear about this work, and I know you'll be excited as well. We're going to hold questions after her talk till you can all come up and talk with her privately, rather than having a general question and answer period. With that all having been said, I hope you can join me in welcoming Linda. Thanks very much.
Well, good evening. Thank you all for coming. It's a great pleasure and privilege to be here speaking to you tonight and to have an opportunity to tell you about some of our research. My laboratory works at the interface of chemistry and neurobiology. We apply the principles and tools of chemistry to understand what, in my opinion, is one of the most exciting and dynamic fields of science right now, namely neuroscience. Neuroscience asks some of the most fascinating and profound questions in all of science. Questions like, how are memories formed, and what is consciousness? Chemists, on the other hand, are like mechanics and architects. We like to take the world apart atom by atom to understand how it works, and then put it back together again and see if we can make it work better. What I show with this slide is that there are multiple different levels at which we can understand or study neurobiology. We can study neurobiology at the organismal level and examine behavior and the circuits that give rise to those behaviors. If we drop down one level, we come to cellular neurobiology. Cellular neurobiology seeks to understand the brain in terms of the interactions between molecules. For instance, we can talk about learning and memory in terms of an enhancement in the communication between neurons in the brain. Finally, there's molecular neurobiology. Molecular neurobiology describes neurobiology in terms of atoms and molecules and the interactions. The discovery of DNA and the molecular biology revolution has made it possible to understand the molecular basis of smell, circadian rhythms, and the structure and function of ion channels that are important for neural communication. What I tried to depict with this arrow here is that there's really a continuum of understanding and a logical progression, and we can go back and forth along this continuum. We can start with an organism and ask if we can understand that organism on a molecular and cellular level. Or we can start with molecules and relate those molecules to their cellular and organismal functions. As chemists, we come into the picture right here at the far end of the molecular spectrum. And we use our ability to create molecules, to synthesize molecules, to understand this larger picture. The hope of this approach is that if we can understand how the brain works at the level of the very small, then that will give us insight into how it works at the level of the very large, and ultimately provide new therapeutic targets for the treatment of aging, injury, and neurodegenerative disease. And before I launch into our science, I wanted to tell you a little bit about the path that brought me here to Caltech and how I started working at the interface of chemistry and neurobiology. As an undergraduate, I fell in love with organic chemistry. I know that sounds strange. Most people have the opposite experience. But I was inspired by my professors at Yale, the late Harry Wasserman and Jerome Burson, who taught me organic chemistry and physical organic chemistry. And what I liked about organic chemistry was that it not only forced me to think outside the box, it also required a great deal of creativity through the design and synthesis of molecules, many of which had never been made before. And so I decided to go into graduate school in organic chemistry, and at Berkeley I joined the lab of Peter Schultz. Some of you know Pete. Pete Schultz was both an undergraduate and a graduate student here at Caltech. He worked with my colleague, Peter Dervin. And so I have Peter Dervin to thank as well. He's my academic grandfather, so to speak. Pete Schultz was famous or infamous not only for his amazing science, but also for his high expectations. He expected his graduate students to work at least 70 hours a week. But I signed on, and honestly, it was one of the best times of my life. I learned how to synthesize molecules, how to characterize enzymes, how to clone a gene, and how to solve crystal structures. And I was surrounded by incredibly smart and talented people. When it was time to graduate, Pete asked me what I wanted to do next, and I told him that I was thinking about a postdoc in neurobiology, even though I didn't really know much about neurobiology. I hadn't taken a formal course in neurobiology or sat in on many lectures. And at that time, there were very few, if any, chemistry professors that were working in neuroscience. Probably my colleague, Dennis Doherty, was the only one. And so it was sort of an intuitive leap of faith, and Pete encouraged me to go for it. He said it was the perfect time and perhaps the last chance that I would have to really learn a new field. And so I Googled molecular neuroscience. Actually, it was Mosaic or Netscape Navigator at the time, not to date myself. And I found a few labs where they were taking a molecular approach to science, to the brain. And that's when I came across the work of Paul Greengard on phosphorylation, the attachment of a phosphate group to proteins and its impact on the dopaminergic system. I didn't know much about phosphorylation, but I did know what a phosphate group was because I was a chemist, and thought that sounded pretty interesting. As it turned out, Paul had trained originally as a mathematician and a physicist before becoming a neurobiologist. And so he didn't mind having a chemist in his lab, and he took me under his wing. Little did I know that at the time, how fortunate I was to join his lab. Paul was such a brilliant scientist that he won the Nobel Prize in Physiology Medicine a year or two after I left his group. And so looking back now, I've been really fortunate to have two brilliant and supportive advisors who were fearless when it came to science. Both Pete and Paul taught me through example that there was no limit to what one could dream of doing in science. And so when I started thinking about what I wanted to do in my own lab, my chemistry background led me to look for molecules that had interesting links to important brain functions, molecules that were not fully understood, but could have interesting and broad functions. One could dream about them. And the molecules that I kept coming across again and again were carbohydrates. When it comes to carbohydrates, if you're like me, you have a love-hate relationship with them. But what I'll show you today is that not all carbohydrates are bad. There are many different kinds of carbohydrates, and they play critical roles in the body. The carbohydrates that we study are not the simple carbohydrates that most people think of, like glucose and fructose that are converted to energy. They're the complex carbohydrates that surround the cells of our body. Our cells are coated with carbohydrates. These carbohydrates form a dense layer, or forest, if you will, that surrounds our cells. Shown here is a surface of a red blood cell that's imaged using electron microscopy. These cells look different from what we often see in textbooks or in movies, like this classic 1966 movie, Fantastic Voyage. And the fuzzy black layer is the carbohydrate coating known as the glycocalyx, which can be up to 1,400 angstroms thick. How many of you have been to California Adventure and taken the Soarin' ride, where you fly around the world in a hang glider? Well, imagine that instead of soaring over the Taj Mahal, that you could shrink yourself down and soar right over the surface of your cells. Now, if you could do that, it might look something like this, where there are geographical features. And now the complex sugars are these mountains and trees, the pine trees and the weeping willows swaying in the breeze. The carbohydrates are like the foliage of our cells. Now imagine next that you wanted to make a landing, that you wanted to make contact with the cell and try to gain entry into the cell. Perhaps it might look something like landing on Endor. The trees, the carbohydrates, are the first sites of contact for communication with pathogens such as viruses or with other cells. And so it's perhaps not too surprising that carbohydrates play critical roles in cell-cell recognition and in the communication between cells. Now, if we zoom in and we look at the structure of carbohydrates, we see that there's a tremendous variety. There's a great deal of chemical complexity in their structures. Now, if I asked you how many bases there are in DNA, you might know that there are 4, A, T, C, and G. If I asked you how many amino acids there are in proteins, you might remember, if you've taken a biology class, that there are 20 naturally occurring amino acids. But if I asked you how many monosaccharide building blocks are used to make the carbohydrates in the human body, does anyone want to call out the answer? Very few people know, and if you do, you're probably from my lab. Depending on whether you count iduronic acid, there are 10 monosaccharide building blocks that make up the complex carbohydrate structures found in vertebrates. Glucose is one of them. And these building blocks are assembled into much more complex structures, some of which are shown schematically here. And they're often attached to proteins or lipids that anchor them to the cell. These are the tree trunks, if you will. What's fascinating about these structures from a chemical perspective is that the carbohydrates, or the glycans as they're collectively called, have structures that are extremely complex and highly diverse. And yet we know far less about them than we know about other biomolecules, such as DNA, RNA, and proteins. The other biomolecules can form only linear structures, like beads on a string. In contrast, carbohydrates can be joined at any one of the 5 hydroxyl groups, which enables them to form both linear and branched structures, such as the complex N-glycans shown here. And the consequence of this is that carbohydrates have much greater structural diversity than other biomolecules. In fact, if you do the back of the envelope calculations, an oligosaccharide consisting of 6 building blocks has the potential to form more than 192 billion structures. Now, this is a huge number, well more than DNA, RNA, or oligopeptides. And for a chemist, this enormous variety of structures is like being a kid in a candy shop. We started thinking about the potential of these different structures to store information and the possibility of understanding their roles in biology. Now, adding to this complexity, to make it even more interesting, the carbohydrate landscape or topiary of our cells can vary from one cell to the next. Each cell type has a different landscape. A neuron looks different from a bone cell, which looks different from a muscle cell. And the glycan structures can change dramatically as we age and with disease. For instance, specific carbohydrate structures are found on certain tumors, and they undergo changes in the levels of these structures, which may be an indicator of that cancer or of other diseases such as Alzheimer's or diabetes. In fact, one of the goals of our research is to develop methods to detect and quantify the carbohydrate structures on individual cells, to count them with single cell resolution. We're working to identify the structures that are associated with learning and memory and with neurodegenerative disorders such as Alzheimer's. And so we can hopefully better understand these complex brain processes on a molecular and cellular level and possibly identify new disease biomarkers. This project is a collaboration between my lab and Leo Pachter's lab at Caltech. You may be wondering, as we often wonder, why does the cell need a sugar coating? Early studies suggested that the sugars might play a structural role. And this is certainly the case in plants where cellulose is the major component of the cell wall. However, we also know that carbohydrates on our cell surfaces can convey important information about the cell and have a profound impact on biology. A classic example is the sugars found on your cells, your blood cells. Now, these are the chemical structures of the sugars that determine whether you have type A, type B, or type O blood. How many of you, raise your hand, are type A blood? Right? Right. If you're type A blood like me, then you have this sugar on the surface of your red blood cells. If you're type B blood, then you have this structure, and if you're type O blood, then you have this structure. Now, you'll notice that the differences between these structures is very subtle. All are composed of a basic trisaccharide core structure, 3 sugars that are linked together. The difference is that type A and B blood have an additional sugar attached. And type A is different from type B blood in that this hydroxyl group has been replaced by an N-acetyl group. This is a very powerful example of how a molecular level change can have a profound impact on human biology. If you're type B blood and you receive a transfusion of type A blood, that's enough to be, that tiny little change of 4 atoms is enough to be fatal. Our immune system is so exquisitely sensitive that it can detect the structural differences between these 2 sugars and reject the wrong type of blood. In addition to determining blood type, carbohydrates mediate other forms of cell-cell communication. They're responsible, for instance, for how many viruses get into cells, such as the influenza virus. They're involved in cancer metastasis as well as inflammation. And 2 areas that we're particularly interested in, they're involved in brain development, both before and after birth, and in the neuronal communication that underlies learning and memory. Now, I just showed you an example of where an atomic level change in the structure of carbohydrates can impact your immune system and have significant implications for medicine. I'm going to turn now to a project in my group where we found that the structure of carbohydrates impacts the functioning of the nervous system, specifically neuroplasticity and the ability of the brain or the nervous system to repair itself after injury. Neuroplasticity is the ability of the brain to adapt and form new neural connections. We know that the connections between neurons in the brain are constantly changing and adapting in response to experience and learning, and that there are critical periods of development when the brain is particularly plastic or malleable. It's been said that the mother of Frank Lloyd Wright surrounded his crib with drawings of cathedrals, determined that her son would become a great architect. And doctors are now suggesting that pregnant mothers play Mozart to their babies while in the womb. These critical periods of development are windows of opportunity when the brain is shaped more easily by the environment to occur not only during development, but also throughout life. However, as we age, the brain undergoes molecular level changes that make it less plastic, less adaptable. For instance, certain molecules can be produced that actively dampen rewiring of the brain. Our lab has been interested in understanding neuroplasticity on a molecular and cellular level. If we can identify the molecules and mechanisms involved, that gives us hope that we could perhaps enhance plasticity or memory and promote neuronal repair in the case of aging, injuries such as traumatic brain injury or stroke, and neurodegenerative disease. Recovery after spinal cord or brain injury, as you know, is severely limited. The neurons fail to grow back. Spinal cord injury is a serious condition for which there are no good treatments. And yet it's a major problem that affects about 18,000 people, new people per year in the United States, typically young men under the age of 30. And unfortunately, the majority of the people live with permanent disability sometimes for more than 40 years after their injury. This was the sad case of Christopher Reeve before he passed away. Now, our lab has been interested in understanding the barriers that prevent regeneration of the central nervous system. It's known that after injury, inhibitory molecules, such as myelin-associated proteins, are released into the site of injury. In addition, chondroitin sulfate, a long sugar molecule, is attached to proteoglycan proteins and is produced by reactive astrocytes to form what's called the glial scar, a barrier to neuronal growth. Importantly, the chondroitin sulfate sugars prevent the nerve fiber from growing back, as indicated by these stop signs in the cartoon. If you degrade the sugars using the enzyme chondroitinase, the nerve fiber can push through and partial motor function is restored in animals. And when we began this project, though, molecular mechanisms were only beginning to be understood. It was unknown how the structure of chondroitin sulfate inhibits neuronal growth, and there was a lot of interest in the field in identifying inhibitory proteins and pathways as potential therapeutic targets. The prevailing view was that chondroitin sulfate served as a physical barrier, similar to overgrown shrubbery that you need a machete to hack through. By treating with chondroitinase, you could clear away that shrubbery and the nerve fiber could push through. And we wondered whether chondroitin sulfate might be playing a more specific role. And as chemists, we saw the molecule as an opportunity to dig deeper and understand the underlying mechanisms. Now, the reason why we thought chondroitin sulfate could be more than just a non-specific barrier was that if we look closely at its structure, we see that it has rich chemical complexity. Chondroitin sulfate is a member of a large family of sugars known as glycosaminoglycans. It's composed of two monosaccharide building blocks that are assembled into long linear chains that can then be sulfated. You may have heard of chondroitin sulfate before. You can get it at Costco. It's a supplement that's sold with glucosamine for the treatment of joint pain. Another glycosaminoglycan that you may have heard of is heparin. Heparin is a multi-billion-dollar drug that's used to prevent blood clotting. And it's been in the news lately because of contamination issues. Fundamentally, all glycosaminoglycans share a common repeating structure, an AB-AB type structure. If nature had stopped there, these molecules might be uninteresting. But there's a twist. Shown here on the left are the monosaccharide building blocks, one of the monosaccharides used to form chondroitin sulfate, which I symbolize here as an orange circle. Each of the hydroxyl groups shown in blue can be either sulfated or not sulfated in nature. And I've symbolized the sulfate group as a red circle. So you can have no sulfation, sulfation here at the 4 position, sulfation at the 6 position, or sulfation at both positions. The net result is that you can have 4 possible structures in the first position. If you consider a simple tetrasaccharide consisting of 4 sugars joined together, you have 4 possibilities at each site. So you can calculate the total number of combinations as 4 possibilities times 4 more possibilities times 4 more times 4 more, or 256 different compounds. What nature has done here is really clever. Nature has started with a simple chemical scaffold and built up diversity by adding sulfate groups. You can appreciate that this could have tremendous implications for the brain, because now we're not talking about a simple tetrasaccharide, we're talking about polysaccharides that can be up to 200 units long. If you consider that each of the positions can be either sulfated or not sulfated, then the number of possible combinations in theory is a number larger than Avogadro's number. If chondroitin sulfate were simply overgrown shrubbery, just a nonspecific barrier, there would be no need for these sulfation patterns. The sulfation patterns would not have been strongly selected over evolution. So there must be more at work here. You may be wondering then, what is the biological significance of having so much potential chemical diversity? And we believe that the sulfate groups encode not genetic information like DNA, but rather functional information. The position of the sulfate groups tell proteins in the brain or the injury site where to go and what to do. And for example, we can take these 2 sulfated monosaccharides and we can combine them in 2 different ways. We can attach the 6-O-sulfated sugar to the 2-O-sulfated sugar, or the 2 to the 6. The result is 2 different molecules, and so you might expect them to have different functions. What we're able to do here is to program information into the molecular structure through the position of these sulfate groups. To make an analogy, we can take a letter and make a word, or we can rearrange them in a different order and make a different word, albeit in some cases with the same meaning. The question is, do the sulfation patterns contain information? Are there words or even sentences encoded in the structure of glycosaminoglycans? The challenge in addressing this question is that in order to unlock the sulfation code, you have to get your hands on the molecules. And the only source of these molecules until recently has been from nature. Traditionally, glycosaminoglycans are purified from an organ, such as the kidney or the brain. You can extract the sugars from that organ and then chemically or enzymatically cleave the long chains into shorter fragments, which you can then separate by size and charge. And there are several problems, however, with this approach. Even though proteins in our body can tell the difference between this molecule and this molecule, our best analytical tools cannot tell them apart, because they're the same overall length and charge. And so the material that you obtain after this long, tedious process is a hopelessly complicated mixture, and you end up with quantities that are often too small to work with. This has made it difficult to relate specific sulfation motifs to biological processes. Our approach is to use chemistry to solve this problem. We can synthesize in the laboratory chemically well-defined molecules or building blocks, and we can link these building blocks in an ordered fashion so that only A reacts with B, only B reacts with C, and so on. The result is we can make well-defined structures in the lab composed of 1 sulfation pattern and 1 length, and we can in principle make any molecules that we choose in large quantities. On the previous slide, I simplified the chemistry behind the process. For the organic chemists in the audience, I'll show you this slide. The chemistry is not easy, but we can control the stereochemistry, length, and patterns of sulfation. In other words, we can make different words and ask what is the meaning, what is the impact of sulfating at different positions. As I said, there's really no other way to access these well-defined sulfation motifs. They're too complicated to purify from natural sources. And so a ground-up, atom-by-atom assembly of these sugars is a powerful way to get at understanding how their molecular structure relates to their function. A long-term goal of my lab is not only just to make a few molecules, but to make all possible sulfated molecules of a given size. For a hexasaccharide, this number turns out to be 4 to the 6th, or over 4,000 different compounds. By systematically comparing the activity of these compounds, we hope to unlock the language and the vocabulary of glycosaminoglycans. So with defined structures, we can address important biological questions. I mentioned earlier that chondroitin sulfate is produced in the glial scar after neural injury, and the chondroitin sulfate sugars prevent the nerve fiber from growing back. The question that we asked was, are specific sulfation motifs inhibiting the regrowth of neurons? To address this, we grew sensory neurons from chick spinal cords in the presence of our defined chondroitin sulfate molecules. We then imaged the cells using confocal fluorescence microscopy. Now here you see the cell body of the nerve cell, which has its nucleus, and then the long main extension, the tail, called the axon. When we treated sensory neurons with molecules that contained a specific sulfation motif, this disulfated motif called CSE, we inhibited the growth of the axons. Neurons treated with the CSE motif had shorter axonal extensions compared to untreated neurons. And we could quantify these effects and observe a dose-dependent inhibition. Now, importantly, when we look at other sulfation motifs, we see no effect. And so these studies demonstrate the importance of the sulfation motif, and we find that the CSE sulfation pattern is a potent inhibitor of neuronal growth. We confirm these observations using a variety of other methods. Here, for example, we used a boundary assay that's designed to mimic the glial scar that forms after neural injury. This is actually a very simple assay where we spot our chondroitin sulfate molecule along with a red dye on a glass slide. We can then add neurons to the slide and watch how they grow and see if they can cross over the sugar boundary. Well, you can see here that when neurons encounter a boundary of CSE, shown here in red, they stop short. They do not cross the boundary. In contrast, neurons can freely cross through boundaries that are formed from these other major sulfation motifs, CSA and CSC. And so what this tells us is that the sugars are sufficient to inhibit sensory axons. You don't need the proteoglycan core protein to which they're attached. And once again, we see that the CSE motif has potent inhibitory activity. Having shown that the sulfation pattern is critical, we next wanted to gain a molecular understanding of how chondroitin sulfate inhibits neuronal growth. As I said, chondroitin sulfate was proposed to serve as a physical barrier, similar to overgrown shrubbery. However, our hypothesis was that the sulfation motifs could interact with specific proteins and direct their activity. Chondroitin sulfate is in fact known to interact with many proteins, and so the ability of specific sulfation motifs to facilitate those interactions and to serve as docking sites for proteins could enable regulation of those proteins and some pretty complex biology. Earlier, I introduced the idea of sugars as the letters of an alphabet. And so the idea is that certain proteins might interact or recognize one word, while other proteins might recognize another word. To make another analogy, these sulfated compounds are like molecular fingerprints. No two are identical. This is very powerful because nature can use one fingerprint to encode one biological function, say cell division, and another fingerprint to encode a different biological function, such as differentiation. Distinct proteins are involved in each of these processes, and distinct sulfation patterns may also be involved. And to add a further nuance to this picture, our studies have shown that the same fingerprint can even interact with different proteins depending on the location and the timing. And so one fingerprint can have multiple different biological functions depending on the context. In the case of spinal cord injury, we thought that the inhibitory CSE motif might interact with protein receptors on the injured cell. And this interaction in turn would trigger changes inside the cell, such as activation of signaling pathways or gene expression programs that would inhibit neuronal growth. The challenge in testing this hypothesis was that there were no protein receptors that were known to interact with CSE. And so we decided to use our molecules essentially as bait to fish out the receptors. We could grind up the neurons from the brain, pass cell extract over our compounds, and then identify the proteins that bound to the sugar using mass spectrometry. And this allowed us to identify many interesting neuronal proteins that interacted with CSE. Among them was a protein called FA4. FA4 is found in the cell membrane, and it's part of a large family of proteins that play various important roles in development and in adulthood. It's known that when these proteins break down and their functions go awry, this contributes to diseases such as Alzheimer's and cancer. And most importantly for this talk, F receptors have been suggested to play broad roles in the body's response to neural injuries. In order to perform their functions, F receptors have to be activated. And they're activated by other proteins called ephrin ligands. These ligands bind to or contact the F receptor, and they induce or cause the receptors to dimerize or become clustered at the cell membrane. This clustering of F receptors, in turn, triggers a series of signaling events inside the cell that lead to changes in the cell, such as remodeling of the actin cytoskeleton or the expression of specific genes. Using a variety of biochemical approaches, we demonstrated that the protein FA4 binds beautifully to CSE sugars and selectively to CSE. And we found that the sulfated CSE structure interacts not only with FA4, but with multiple other inhibitory neural receptors. So CSE serves as a docking site for many proteins. Now, as chemists, we like to understand how and why things interact. Understanding the interactions between molecules at an atomic level is important for modulating those interactions and controlling biological processes, which is the end goal for many pharmaceuticals. And we wanted to figure out how CSE was being recognized by the FA4 receptor and why that interaction was so selective for the CSE fingerprint. To answer this, we collaborated with Professor Bill Goddard's group here at Caltech, who's sitting in the front row. This collaboration was initiated by a student of mine, Peter Clark, who's now a professor at UCLA. And I like to tell people about this collaboration and how it got started because it's a perfect example of what makes Caltech so unique. First of all, we get amazing students, but I think anyone who watches The Big Bang Theory knows that. We get brilliant minds, right? Like Sheldon Cooper, or in my case, Peter Clark. When Peter joined my lab, he came from Cornell University and he had 6 degrees. He had earned 6 bachelor degrees in 4 years in chemistry, biology, mathematics, economics, philosophy, and physics. And so he was no slouch, and when we sat down and we started talking about what he wanted to do for his thesis project, he said that he wanted to have some computational chemistry as part of his thesis. And so I told him about our glycosaminoglycan project and suggested that he go over and talk to Bill. And from that conversation began a long-standing collaboration with the Goddard Lab that continues to this day, nearly 15 years now. I think if you ask anyone on campus, they would agree that the sense of community and the low barrier to interaction here at Caltech is truly unique and enabling. And we have a special culture that gives our graduate students the freedom to explore their own ideas. What Peter and later Claude Rogers discovered, along with Adam Griffith from the Goddard Lab, was that CSE interacts with 2 sites on FA4. Each site is lined, interestingly, with lysine and arginine amino acids. This is significant because these amino acids are positively charged residues, and they're positioned to align perfectly with the negatively charged sulfate groups on the sugars. There's a beautiful combination of both electrostatic and van der Waals interactions that help us to understand why and how this protein recognizes CSE. Using site-directed mutagenesis, we could exchange those positively charged residues for neutral, uncharged amino acids. And when we made this mutant protein, we see that it essentially abolishes the interaction with CSE. So this confirms the binding site, and I'll come back to this mutant later because it gives us a very useful tool. Now, I mentioned that F receptors become activated by other proteins, the Ephrin ligands. And activation occurs because the ligands can induce dimerization or clustering of the receptors. Now, here you see the crystal structure of two F receptors, one in pink and the other in green, that are clustered together by two Ephrin ligands in yellow and blue. Now, when the receptors come together, when they cluster, we observe something interesting. The binding sites for CSE come into close proximity. I've circled the adjacent sites here. So this suggested to us that a long sugar chain might be capable of spanning two or more receptors and engaging possibly both receptors to induce dimerization or clustering. Now, sure enough, Greg Miller, a grad student in my lab, found that CSE polysaccharides could indeed cluster and activate FA4 receptors. When we image the receptors, we see that they're distributed broadly and uniformly across the cell surface, as you can see here in yellow. However, when we add a protein ligand, Ephrin A5, remember Ephrin A5 causes the receptors to cluster. And as expected, we can observe that clustering as indicated by these yellow puncta. What was even more exciting was that when we added CSE polysaccharides to the cell, we see the same kind of clustering and activation of the receptor that we see when we add the Ephrin ligand. That's exciting to us because this is the first example, to my knowledge, where a carbohydrate has been shown to control the activity of F receptors. Until now, it's been all about protein ligands, not about the sugars. And so this suggests a novel mechanism for regulating the activity of a very important family of proteins, none of which had been linked to chondroitin sulfate before. In fact, we think that the regulation of the Fs by CSE goes beyond FA4. Of the 14 F receptor family members, at least 6 of them interact with CSE. And in other studies, Claude Rogers in my lab showed that a related protein, FB3, plays an important role in the ability of CSE to guide retinal axons and form neural circuits during brain development. So this chemical approach is revealing new mechanisms that underlie important biology, not only related to neuroregeneration, but to a host of other functions. It's also worth mentioning that the way in which chondroitin sulfate engages these receptors at the cell surface was unusual and unexpected, at least to us. It was known from many elegant studies that chondroitin sulfate and heparan sulfate glycosaminoglycans can recruit soluble proteins, such as growth factors, to the cell surface and assemble ligand receptor complexes at the cell surface. But in addition to this well-established mechanism, our studies indicate that chondroitin sulfate sugars can also engage cell surface receptors directly, even in the absence of their canonical soluble ligands. And by controlling the clustering of these receptors, they can regulate biological processes in the cell, such as inhibition of neuronal growth. So thus far, I've shown you that CSE interacts with receptors such as FA4 to inhibit neuronal growth. But does this interaction play an important role in the ability of injured neurons to grow back? To test this, we use microfluidic devices that separate the cell body of the neuron from its axons. Just to remind you, the axon extends from the cell body and it forms connections with other neurons. So stimulating the regrowth of axons is essential for neural repair. And we added the neurons to the left-hand side of this device and then allowed the axons to grow through tiny microfluidic channels into the other side. Well, the axons are shown here in green. You can see that they have grown through these microfluidic channels into the right-hand side. After 5 days, we performed an exotomy. We severed the axons by applying a vacuum to the right-hand side. Next, we watched those axons grow back. Okay, so now in the absence of any inhibitory molecules, you can see that some of the axons can grow back within 24 hours after the injury. But when we add inhibitory CSE molecules, the axons fail to grow back, okay? And that's quantified here in red. So we now have an excellent assay for axon regrowth after injury, and we can use this assay to evaluate the importance of the CSE-FA4 interaction. Now specifically, we took neurons that had either normal FA4 receptors or mutant receptors. You may recall that when we replaced the positively charged amino acids in the protein with neutral uncharged amino acids, we obtained mutant proteins that could no longer bind CSE. Now the prediction is that if the CSE-F interaction is important for inhibiting axon regeneration, then neurons expressing the mutant receptors should show enhanced regeneration because they can no longer be inhibited by CSE. Now sure enough, this is exactly what we saw. Neurons expressing the mutant receptors showed greater axon regrowth relative to neurons expressing the normal wild-type receptors. So this tells us that the CSE-FA4 interaction is inhibitory to injured axons. And importantly, when we block this interaction, we can stimulate axon regrowth. With this information, we have an opportunity to intervene in this process. If CSE inhibits axon regrowth, then blocking this motif may promote neuronal repair. To block the sugar and its interactions with inhibitory receptors, in this case we generated a monoclonal antibody against the CSE tetrasaccharide that we had synthesized chemically in the lab. Because we used a synthetic molecule that had a well-defined structure instead of the polysaccharides, the natural polysaccharides that were heterogeneous, we could obtain antibodies that were highly selective. This antibody, as you can see, does not recognize any other sulfation motifs or glycosaminoglycan classes. I'm going to skip directly to in vivo studies using this antibody. We chose an optic nerve regeneration model, which was performed by our collaborator, Dongfeng Chen at Harvard Medical School. Now, this is a classic regeneration model in mice where the optic nerve connecting the retina to the brain is cut. It's crushed, and then we can monitor the ability of the optic nerve to regenerate. And we first wanted to see if the CSE motif is upregulated in response to this injury. Shown on the left is the control tissue, the uninjured tissue. The CSE sugars are labeled in red, and you can see that they're present, albeit weakly, in the sample. On the right-hand side is the injured tissue. You notice a much stronger red staining, indicating that CSE is in fact produced in response to this injury. We also observed a dramatic upregulation in other experiments of CSE after spinal cord and cortical brain injuries. The next question, the key question, is will blocking the inhibitory CSE motif promote neuronal regeneration in vivo? We delivered the CSE antibody or a control IgG antibody into the injury site, and sure enough, application of the CSE antibody stimulated regeneration. We observed a 6-fold increase in the number of regenerating axons, and the axons extended twice as far as in the control group. Importantly, the extent of regeneration was comparable to that seen with chondroitinase. So blocking the CSE with this antibody is as effective as complete removal of the chondroitin sulfate chains. This highlights the inhibitory potency of the CSE motif in vivo. Furthermore, we found that the antibody could be combined with other treatments to stimulate regeneration even further. We used the cyclic AMP compound to stimulate the intrinsic growth of neurons while simultaneously blocking the inhibitory CSE environment using the antibody. And this combination of treatments nearly tripled the distance of regeneration. And so we're really excited about these results. They demonstrate that a specific carbohydrate structure plays a critical role in limiting neuroplasticity in vivo, and blocking this structure offers a new promising strategy for neuronal repair. Because CSE interacts with multiple proteins, blocking the CSE motif may be more effective than inhibiting individual proteins or pathway. This is a strategy that's been taken by some pharmaceutical companies. In addition, blocking CSE could provide an attractive alternative to chondroitinase. Chondroitinase ABC is currently in clinical trials for spinal cord injury in dogs. However, there have been challenges with translating this bacterial enzyme into the clinic. For instance, chondroitinase can be immunogenic, and it has to have repeated local injections because of its thermal instability and short half-life. So the CSE antibody may offer a viable and effective alternative. We're now testing this antibody in a clinically relevant spinal cord injury model in collaboration with Liz Bradbury's group at King's College London. Liz is assessing the ability of the antibody to promote axon regeneration and restore upper limb and skilled hand function in rodents. And we've also generated a single-chain variant of the antibody that has improved in vivo properties, and we've been exploring various viral delivery strategies in collaboration with Viviana Gradinore's group here at Caltech. As a complementary approach, we've also developed small molecules that inhibit the sulfotransferase enzymes involved in the biosynthesis of CSE. These enzymes attach the sulfate groups onto the sugars, and so they are another powerful way to block CSE. Finally, in addition to spinal cord regeneration, we're examining the potential of these approaches to promote the remyelination of axons, which could be exciting for multiple sclerosis, and the potential to enhance neuroplasticity and memory formation in mouse models of neurodegenerative and psychiatric diseases. I won't have time to tell you about our work in that area, but Tracy Huang, a postdoc in my lab, has recently discovered that the CSA motif plays a role in regulating the balance of excitatory and inhibitory synapses in the brain and the formation of social memories, our ability to remember acquaintances. And so we think that these findings could have important implications for neuropsychiatric disorders such as autism and schizophrenia. And this is an area that we're very excited about understanding in the future. To conclude, I hope I've shown you that carbohydrates are more than just a source of energy or something to be avoided in your diet. We've shown that carbohydrates such as chondroitin sulfate play important roles in neuroplasticity and axon regeneration after injury. Blocking these sugars may open up new opportunities to enhance plasticity and promote neuronal repair. Most importantly, I hope I've shown you the potential for chemistry to enable new insights into neurobiology. Caltech is well known for fostering interdisciplinary science, and I'm thankful to be here where we can cross traditional boundaries and go back and forth along this continuum. Finally, the most important slide of all, I'd like to finish by thanking all of the students who did this work, both former and current members of my lab, including students that have worked on different projects that I didn't have the time to talk about today. The credit really belongs to them. I'm just the spokesperson. They're the ones who did the hard work. I also want to thank these funding agencies, both federal and private, that have supported this work, including the Chen family for recently supporting a grad student of mine to work on the project in the Pactor, with the Pactor lab. Finally, I want to thank you all for coming tonight. I'd be happy to answer any questions. Please feel free to come up to the stage afterwards. Thank you.